Why use a standard at all? The ZymoBIOMICS Standard is a defined mock microbial community with two versions, a whole cell mix and a purified DNA mix. The idea is simple. The whole cell control checks whether your lysis and extraction are doing their job. The DNA control looks at what happens after that, so it reflects PCR, sequencing, and bioinformatics. Use them together and you can see where bias is creeping in, not just that it exists. Microbiome work lives and dies on controls. Bias can slip in during lysis, DNA extraction, PCR setup, on the sequencer, or later in the pipeline. Without a reference, it's hard to tell if your community profile reflects the sample or your method. That's where the standard helps. It gives you an outside yardstick so bias becomes visible and fixable. In practice, we run both controls. The whole cell mix includes organisms with different cell walls, so if your lysis is too gentle, gram-positive species may show up undercounted. You don't have to guess, you see the miss right next to the expected composition. The DNA standard removes lysis from the equation. If results drift here, it points toward PCR preferences, sequencing quirks, or how your pipeline is classifying reads. Seen together, the two controls narrow the problem to specific steps, which is usually what you need to decide what to change. QC reporting makes this tangible. You compare expected versus observed and look at the gaps. A few bars sit lower than they should. That may suggest incomplete lysis or an extraction chemistry that favors certain genomes. A GC-dependent pattern appears. It's likely a PCR or mapping issue. You tweak, rerun the control, and check whether the bars move toward the expected profile. It won't be a perfect match, mock communities rarely are, but the trend should improve with each fix. What do you get out of this? Faster troubleshooting, results that hold up when you repeat the study or scale to more sites. Safer comparisons across cohorts and instruments. You're separating biological signal from method noise, which is really the point. A quick example. One group kept undercalling high GC gram-positive taxa. The whole cell standard flagged the extraction step. They adjusted lysis time and detergent, then repeated the run. The follow-up chart lined up much closer with the expected mix. Not perfect, but clearly better, and the improvement carried into their main dataset. Ready to try it? Contact Nano Diagnostica. We'll help you get started and answer all questions.